2, 1.
Good afternoon, everyone. I'd like to welcome you to the live Q&A session for theme number 1, the science of infectious disease. As we get started this afternoon, I'm going to do a quick roll call just to confirm that our speakers are live and available. So, Dr. Ewing, are you online, and can you confirm you can hear me?
Yes, hello, good afternoon.
Thank you. Dr. Dexy Chen, are you online, and can you confirm that you can hear me?
Yeah, I can hear. Good afternoon.
Thank you. Dr. Sujatha, are you online, and can you confirm that you can hear me?
Yeah, loud and clear. Thank you.
Oh, loud and clear. Thank you. And Dr. Shemelak, are you online, and can you confirm that you can hear me?
Yes, I can hear you.
Oh, fantastic. I want to thank all of our presenters today. They presented some fascinating work, and I'd like to get started with the Q&A. So the first question is for Dr. Ewing. Excellent work identifying the adaptive ER stress and reduced metabolic function of the TCA cycle and the baseline conditions that contribute to an individual's progression to developing symptoms post-infection with the yellow fever vaccine. You discussed several possibilities that could potentially be driving the subset, such as BMI, genetics, age, microbiome, etc. As you look to what we've seen with COVID clinical presentations thus far, can you comment on where you'd like to see the field focus their efforts?
Thank you. That's a really good question. I'd love to kind of understand whether this ER stress and metabolism have roles to play in the pathogenesis of COVID-19. Obviously, the challenge there is going to be recruiting the cases before they're infected and trying to understand what is that baseline state, both in terms of the ER stress as well as metabolism. That would be a major challenge, and maybe in household clusters or in nursing homes or dorms that may be possible, but I think beyond that it may be very difficult. But the key to that would be to try and recruit the cases before the onset of symptoms, ideally even before infection, and that's hard. And that's why in some ways the vaccine gives us that opportunity to standardize when infection actually occurs. Another way of coming at that is just to use drugs like metformin, which obviously modulates ER stress, as a way of asking whether that would have any impact on changing the course. But those are there's quite a lot of work to be done to address all those questions. Thank you.
Thank you. So the next question that's come in is for Dr. Dexy Chen. Can you talk about one of some of the next projects that you have coming up, looking at elevating CD8 positive T cells in the immune system of severe COVID-19 patients? So what's next on the horizon for you? So, Dr. Chen, are you still with us?
You mean double positive T cells? Can you repeat your question?
Yeah. How about I'll rephrase it slightly. So maybe you can talk about what's next up for you in terms of the projects you have coming up when you look at the immune system of severe COVID-19 patients. So what would you like to see as the next progression of your projects?
I think our next project mainly focus on the molecular mechanism. So we run more than 200 genes in the coronavirus 19 the patient samples the PBMC. And I think we will find the more cell signal pathway how affected the double positive T cell or CD8 T cell or TGF beta positive T cells, how deregulation to affect the corrective or severe the COVID-19 virus disease process. Thank you.
Thank you. Dr. Locke, this next one is for you. You've done some really nice work with the different morphologies of the dengue virus. You suggest treatments or vaccines that incorporate multiple morphologies. As you commented, they are all infectious. Can you comment on the relative infectivity of each morphology? And then a follow-up question, or actually I'll let you answer that one first, and then there's a follow-up question.
Okay, so for the dengue mature and dengue immature, their infectivity is similar. So for the immature virus, because they infect a different cell completely, so I cannot compare directly, but they can infect by different pathways, like binding to DC sign positive cells, and also their infectivity can be assisted by antibody, because antibody can help them to bind to Fc receptor on macrophages, which are the target cells. And for the club-shaped particles, because there's only 20%, like a plug assay, they shows you in terms of logs, right? And you won't see much in terms of logs. So, but their ability to attach to the virus is about the same, and as the mature virus, and also they contain RNA, so we think that is infectious too. Yeah.
Okay, thank you. So this is a question actually for the group, and so maybe we could take turns or maybe have one or two answers. But in terms of, can some of you speak to your collaboration opportunities with other researchers? How has the pandemic impacted your ability to collaborate? Has it increased? Has this, talked about different partnerships that have perhaps developed and given you greater opportunities?
I guess I'll be happy to start. No, it actually has opened up a lot of opportunities. I mean, I don't normally work on coronaviruses, but I guess now it's kind of like all hands on deck situation, and so we do what we can, and the collaboration that we have with Arcturus Therapeutics is actually born out of initiative, a tie-up that the Economic Development Board of Singapore, initiated between us and Arcturus Therapeutics, and that's how it came about. And prior to this this pandemic, I had not thought about working on mRNA vaccine. So it's one of those great opportunity came along. It was too good to pass. So I think there's many more such instances, I'm sure. Thank you.
Fascinating. Actually, has anyone else had an opportunistic way for the pandemic, that in terms of creating a new way to collaborate or work across disciplines or public or private space? I'd like to hear maybe one of our other speakers comment on this.
By my personal experience has been that this pandemic has opened doors of different groups of researchers, be it chemists, be it vaccinologists, basic researchers, who all have come together to join hands to understand the disease in a more holistic manner. And that is very important in tackling any disease, which I have been seeing. So even though it has presented more opportunities in terms of different kind of collaborations, which we would not have been thinking about at other times, what is important is to maximize this opportunity in disease management as well as understanding. I think artificial intelligence have come up in such a big way in the last 7 months that it hasn't done in the couple of years, except for those experts who are actually working on that. So this is the time which I feel that we need to maximize the partnerships that have come from different group of experts in tackling infectious diseases in general and, of course, COVID in particular. Yeah.
Thank you. Actually, just building on that comment, we've had a question in as a proactive approach. What has been the use of AI within predicting future viral strains that can cause this pandemic? And what do you see for that either in your work today or in the future?
Yeah, so I was telling that, like even when I told in the presentation, there's a lot of noise that's present right now, and that is something we really look into it. We need to de-learn the information that we've got, sieve out the good information, and then use it for predicting or understanding what could be the new re-emerging or new viral strains that has been unheard of, in at least in human population, to study what kind of evolution that has been happening in these viruses. It could be sylvastic species, it could be viruses that have been largely presented only to the animal sciences. But yes, we can definitely make use of artificial intelligence to understand what could be or predict the kind of novel viruses that could be coming in the future of infectious diseases. Yeah.
Thank you. So I have another. Oh yeah, absolutely. Please go ahead.
I was going to say thanks. I totally agree with that view that I think we now have great tools like artificial intelligence that could help us with our work, troll through the public databases and literature to help us make more educated guesses on what to expect next. But I also think that in these publicly available information, there's also a lot of noise that it'll be very difficult to see through sometimes without experiments. I think you kind of need both at this point in time. I don't think that artificial intelligence and all that is only as good as the data that's fed into it, and we still need better data. Thank you.
So I actually thank you. That's it's a great sort of pivot in terms of speaking about the data. I'd like to pose this question to all of our speakers, as it touches on your research and all of your research in some respects. But can you comment on the recent reports of COVID reinfection?
I can start, I guess. Yes, I don't think a reinfection is at all surprising. Obviously, the question is what is the risk of severe disease relative to the first infection, and whether that then constitutes a thing to worry about for the future. And I think for that, the case reports that's coming out is not clear enough. Personally, I've not seen any data apart from the and I know that there's more than one case now being suggested across the world. So it'd be good to see the data and understand what's the difference between the first and second infection. But at least insofar as the first case report on this subject, it does look like the adaptive immunity that was generated from the first infection seemed to have protected the patient from a symptomatic second. And there maybe the T cells be playing a very important role, because from what I can gather was that the subject had sub-detectable antibody levels, and maybe that was why the person got reinfected. But with the T cells, then perhaps that's why the infection became asymptomatic. So it'll be important to see this, the evidence for all these other reinfections that are beginning across the world, and understand whether they're, first of all, truly a reinfection or a misdiagnosis. Thank you.
So we
have been seeing cases. Oh, go ahead. Yep, go ahead. Thank you.
Yeah.
Please, no, go ahead.
There have been cases of reinfection or reports of reinfection in India as well. So there are a couple of things we need to look really closely when we assign a case as reinfection. One is whether after the first time, whether they have developed antibodies or if they're still negative for antibodies. And the second thing is, the second important aspect is what was the real-time PCR data which looked like? Was it qualitative? Was the viral load known? And if it was qualitative, what was the CT value? If you're going to have a very high CT value, which speaks about very less viruses, whether that or whether it was actually a reinfection or the virus present, hidden maybe it was in the reservoir and then it comes up at another later stage. So these are the few aspects that we need to look into on an individual case basis before we actually assign a specific case as reinfection. So we still need to do a lot of work before we actually use this term and then look into the science behind it, because right now the important aspect is how are the clinicians going to deal with the whole system. The biology comes later. And these, if you're going to have a metrics for the clinicians to define a reinfection versus a recrudescence or it has been, so these are important aspects we need to define first before we go and claim that yes, there has been whatever we see as reinfection is truly a case of reinfection. Thank you.
Thank you. So I'm just making sure I go through the questions, and so we have a couple more coming through here. And then when I go through here, I think that so this is a question actually that just popped up in terms of Dr. Locke. Do you agree that better understanding of the human microbiome will help researchers to come up with alternative approaches to handle viral infection?
Is that for Dr. Wee, I think? Not Locke.
Shami?
Oh, is that me? I don't study human biome, microbiome. It must be for someone else. It's a mistake. Yeah.
I'm just reading the question here. My apologies here. Do we have someone else who would like to take that one?
Is that a question on human microbiome? Is that what it is?
Yeah, I think so.
Oh, okay. Yeah, I'm happy to take that. It's a great question. No one really knows right now. I think the human microbiome is a hot area of research. Obviously, we know that the microbiome influences the immune response in various ways, right from the from innate immune response all the way through to the idea that maybe even influences blanking out a term for it. But anyway, the innate immune response does undergo some epigenetic changes, and it influences, and microbiome may actually influence that process, and that then affects how it responds to other acute infections. But I don't think it's well understood right now, and it's hard to kind of interrogate it clinically. Difficult to change people's microbiome steadily across time so that you can understand its impact on health with regard to how we respond to an acute infection. Thank you.
Thank you. Okay, so I'm not going back to Dr. Locke. In terms of looking at the underlying protein structure, and you spoke a lot about the E1 protein conformations between the smooth and the spiky viruses in regards and changes to temperature. Can you comment on other potential physiological factors that you think might be contributing?
So right now, the temperature is the most obvious, but I think the pH also affects. So we kind of know, at least for dengue virus 2, at pH 8, the virus surface is tighter, and then as you go lower, pH 7 point something, the surface become a little bit looser. So the pH also kind of matter. Yeah. But every strain is different because some strains are really very tight. I mean, they're so tight that when you run a SDS page cell, the E proteins, remain associated with each other. We see like monomers, dimers, trimers, tetramers, and the higher molecular weight proteins. So, yeah.
Yes, thank you. Okay, we have another question here for Dr. Ooi. And then this is back to, with COVID-19 typically taking 5 to 6 days passing post-infection before we're able to capture them clinically, so the inflammatory response is already at play. And it's really quite genius to using the live yellow fever vaccine to understand the implications of the baseline contribution. What differences between yellow fever and COVID-19 infections do you expect or predict?
Great question again. Thank you. I guess, from what we have been able to see from the yellow fever vaccine, I think all we can say is that, the likelihood that those people who have underlying adaptive ER stress and lowered Krebs cycle activity, upon infection, because an infection then requires both protein production, which would exacerbate ER stress, and energy. Therefore, it tips the cell over into a maladaptive ER stress response, and that then leads to symptoms. And I think that's all that we can say about it, because beyond that, what then leads to severe disease, I think it's viral burden dependent. Right? In the case of dengue, yellow fever, it will be viremia because you can measure that in the blood. But COVID-19, maybe not, because the viral burden, the SARS-CoV-2 burden, is in the airway, and likelihood the lower airway, which is not easy to sample. And so I think the subsequent inflammatory process that then differentiates those who get mild disease versus those who get severe disease because of the inflammation, I don't think is ER stress driven. At that point, I think it's virus driven. So I think the ER stress can only go so far as to explain why some people do not get symptoms even though they're infected with the same virus. So the progression to severe disease, I think it's a whole different layer of biology added on to this fundamental differentiation between symptomatic and asymptomatic infection. Thank you.
So we have another question coming in here, and in terms of if a COVID-19 vaccine is made available, is there which target group is considered important to receive the vaccine, and is there not just one target group, but are there multiple groups that we'd like to target? And I'd like to open this up for discussion across the group.
I'll be happy to start. So I mean, that's a great question, and I suspect that that's something that would be different from country to country. Obviously, you definitely want to target the vulnerable group right from the outset. Those, the elderly, those with comorbidities, the people who are more susceptible to severe disease with SARS-CoV-2 infection. However, that also has to be balanced with what we will eventually be determined through phase 2 and possibly 3 clinical trials, because the vaccines in general don't work very well in the elderly. And so if that's the case, then vaccinating the elderly may not be the most efficient way of applying the vaccine. We may instead want to vaccinate those who care for the elderly, so family members, staff of nursing homes and all that, and the healthcare workers who look after these elderly patients. So it really, I think, and then of course in some countries where there is higher rates of hospital-based infections, then it would definitely make sense to vaccinate the healthcare workers, and then after that the population. But I think it depends on country to country. For instance, in Singapore, we have had hardly any. I think there were a handful of healthcare workers who were infected. But by and large, the problem was actually mainly in the community, especially in the migrant worker dormitory. So you've got to have to vaccinate where the problem actually is. Thank you.
Before I ask the next question, would any of our other speakers like to comment as well? Just in terms of the question or further add their opinion to that answer?
Well, I think In-Yong is right. We should vaccinate, the elders, so because they are the ones that could be more endangered, by this COVID infection, and likelihood of severity is high. And also people in the first line, like, those who work in the hospital and grocery stores, and those who interact with lots of people.
Okay, so we have another question coming in here in terms of pandemics normally follow a common pattern, and we hear that many of the pandemics of the past have had wave 1, 2, and sometimes 3. With respect to COVID-19, is there any information on which wave is currently in progress in specific countries?
I guess I don't know about the other speakers, but what I know comes from the news as well. So if anyone is reading the news, you'll get the updates. And I mean, to me, it looks like France is appearing to have another. The cases are looking to have an exponential growth again. And I suspect that this will apply to those countries that relax their control measures a little bit too much, perhaps. And so, I think we should expect a second, we should expect a third wave until a vaccine becomes available. I don't think this virus is going to go away at all. It's become too entrenched in the human population. Too many people already have it and carry it, and it can be easily transmitted. Right? But it's not as easy as not as highly transmissible as, say, measles or anything, but nonetheless, the estimated basic reproductive rate of R0 is about, somewhere between 2 to 4 or around 3. It's still large enough to be of concern. So I think the tricky part is how do we maintain this kind of social distancing and control measures, mask wearing and all that, when, first of all, it's not ingrained in the way most of us were brought up, and it's very counter to human psychology to some extent. So it's hard to bear, but until a vaccine becomes available, this is the only way we can stop or reduce virus transmission.
So actually, I have a question. So I heard some report also from the news that some Italian doctors and Spanish doctors say that the virus has gotten weaker because there are less severe cases in their hospital. I mean, is that true, or is just that those with severe cases already have gotten the disease, and so the leftovers are the strong ones, and therefore you don't have much severity? I mean, the severity cases are lower.
From my own impression was that the initial wave in Italy affected the elderly, and that's why there were so many deaths. And I think now it's affecting mainly the younger adult population, and so the disease outcome seems to be a lot better. I don't think that the virus has changed sufficiently to be able to make such a difference in the outcome. Obviously, there are mutations, and our colleague Gavin Smith found one deletion in the genome in Singapore actually, but it doesn't appear to make a big difference to the outcome clinically.
At least with respect to the genome information, there is nothing to indicate that the viruses have changed substantially to have a difference in their pathogenicity.
That's right.
So with respect to the virus, there hasn't been any change.
Okay, thank you.
Excellent. All right, you guys, we're at our time, and then we could continue this conversation for many more minutes and many more questions. We do have more Q&A coming through. Unfortunately, we're going to have to go to the next session, but what I'd like to do is thank everyone in our audience for engaging and submitting questions, and all of our speakers on our panel for their presentations and for their dialogue and for their conversation today. We really appreciate it, and just really want to thank you for your time. And so especially this concludes our first session, our theme, the science of infectious disease. The content will be available post-event on demand through the Labroots platform. And please stay with us for our second theme this afternoon, the economics of disease control, considerations for public health. Thank you, everyone.
Thank you.
Thank you.
Thank you.
Thank you.